Okay, so I'm Emily Krause, and I'm a brand new PhD student in John Spear's lab here at Lions. I started about 3 days ago, so I haven't really started research. Eventually, I will be working on a NASA Astrobiology Institute grant, doing characterization of the subsurface microbial communities in Oman. So that's exciting. But today I'm here to introduce Tom Schmidt. And Dr. Schmidt got his PhD in microbiology at Ohio State University in 1985. And then went on to do a postdoc at the Scripps Institute of Oceanography in San Diego, California. And in 1987, Tom took on a postdoc with Normposium in Indiana University, and he's going to talk about that a little bit today, I think. But during his long and distinguished career, which I can't fit on this little card here, Tom was also a co-director of the microbial diversity course in Woods Hole, Mass., which I think a lot of you have gone through, and I hope to go through someday. But currently, Dr. Schmidt is a professor in both ecology and evolutionary biology, as well as internal medicine at the University of Michigan, where he's a special joint position there. And he now focuses on research into the human gut microbiome, still using techniques he learned in Normposium's laboratory. So please welcome Tom Schmidt.
Thanks very much, Emily. That's pretty good. Three days ago you start and you pressed in the service right away. I appreciate the invitation. Hey, what a pleasure it is to be here to celebrate Norm's contribution not only to science but to all of us and many other people who've passed through the laboratory. I thought that rather than talking about the microbiome, which we're working on a good bit today, that I'd share some results and some ideas about a project that really had its inception while I was a postdoc with you, Norm. And that has to do with ribosomal RNA operons and observation that those are often repeated in bacterial genomes. So as Norm mentioned last night, the 16S, 23S, and 5S ribosomal RNAs are typically in bacteria and archaea organized into an operon with one or two promoters, a terminator. Occasionally there are tRNAs sprinkled in internally transcribed spacer, either at the beginning or at the end of the operon. And that operon is repeated around the genome. And in E. coli, it's repeated 7 times. So it doesn't matter which of the strains that Gary talked about. Lots of their gene content is coming and going. But the ribosomal RNAs appear not to be. And in fact, they're in the same position in the E. coli genome as pictured here. Remember, this is in a background of primarily single-copy genes. So most genes in bacteria are present in a single copy. Sure, there are examples of genes with 2 or 3 copies, but the real exception to the rule are the ribosomal RNA genes. And this seems to be a characteristic that is consistent at a species level. That is, wherever you get E. coli from, it has 7. Other species have 1. Or 2, or in Vibrio, 15. And so, why is that? And what can we learn about the bacteria by thinking about the forces that are maintaining these multiplicity of ribosomal RNA operons? So that's what I'd like to share some results and ideas about today. But before I get there, I'd like to step back to where these ideas came from originally. And so Norm mentioned this article that was in ASM News in 1985. This has really revolutionized microbial ecology. I just came from the ASM meeting in New Orleans, and I don't think I went to any session where this general approach, that is of reaching into nature and extracting DNA or RNA and doing sequencing from it, I don't think I went to any session where that wasn't incorporated as part of it. It really has had a revolution. It's really been a revolution in microbiology and in microbial ecology, and it's more than who's there, right? Because what they mentioned at the beginning is that one can look at the relative abundance of unique sequences, and so you can do ecology. You can look at temporal and spatial patterns. You can look at invasion of communities. And so this really sets the stage for lots of what's happening in microbial ecology today. I read this article in 1985. I was a postdoc out at Scripps Institute working in Ken Nielsen's lab. And I read this and was persuaded completely that I had to do this. And so after a few days, I got the courage to give Norm Pace a call. He answered his own phone. I introduced myself and said, look, I read this article, and I should be interested in the postdoctoral position. He said, I just hired Ed DeLong for that position. He said, I don't have one. I mean, I was crushed. I was disappointed. But I kept an eye on it, and about a year later, there was an advertisement for a postdoc position. I applied along with lots of other people, I'm sure, and was delighted to join the lab. So when I joined the lab, at least in those days, I did what everybody else did, and that was learn how to do reverse transcriptase sequencing. So you could pick whatever organism you wanted. I was just coming from a lab and studying luminescent bacteria, Photorhabdus luminescens, that's symbiote of nematodes. And it seemed reasonable to ask whether or not it was phylogenetically related to the luminescent bacteria that are found in lots of marine organisms. And this was a terrestrial nematode that Photorhabdus was symbiotic with. And so that was the first assignment, okay, sequence the ribosomal RNA. So I set out to do that, and it was my first gel. So, I also learned at the time that Norm has an uncanny ability to show up when the results were coming in. So I was looking at this gel. By the way, maybe it doesn't matter if you turn the lights down or not. And there are bands there. Anyhow, I was looking at this gel, and I sensed the presence. Norm was looking over my shoulder, and I tried to make the best of it, hold it up to the light a little bit. I mean, there wasn't much to remedy it, and so I mean, so I just waited for Norm's assessment of it, which he gave, and he said, "Tom, he said, that's really dog shit." So, I mean, it started not a very auspicious beginning in the lab, but I'll have to say, I mean, Norm, we sat down, and he said, let's go through your notes. What did you do? And I think it was magnesium and NP40. So I knew I was in the right place, and I was delighted to be there. He was somebody that not only had the big picture in mind, but had his hands involved enough to be able to sit down and go through protocols and help you learn how to do these techniques. So that was my beginning in the PACE lab. Also at the time, there was some interesting interaction going on between the RNA biochemistry and the search. And Alex Bergen was in the lab looking at the excision of intervening sequences from the 23S ribosomal RNA and salmonella. Right, and as you can see here, here's E. coli, the intact 23S ribosomal RNA and 16S ribosomal RNA. And what Alex and company showed was that that's not necessarily the case in all bacteria. And in fact, it's even different in different species of salmonella. So in this species, there's some intact 23 and 16S, but the other ribosomal RNAs, which have an intervening sequence, the RNA is excised, and there are differences amongst these 7 ribosomal RNAs. In Typhimurium, evidently all of the 23S ribosomal RNAs have an intervening sequence. So I show this because at the time, I mean, it just reminded me that when there are multiple copies, they're not all the same. And in this case, they have intervening sequences. But if you look at the sequences themselves, there are differences also. So here's a maximum likelihood tree of the 7 16S ribosomal RNA genes in Salmonella enterica. These tick marks here, I just put there so that you can see that there are 5 different genes there. They're identical. No differences in the 16S genes. 2 of them have some differences. In E. coli, this is K12, I think. 3 of the genes are identical. A few have a handful of differences, and one's a little bit more divergent. So, I mean, this struck me while I was there, and it's something that I've pursued on the back burner for well, ever since I left Norm's lab. And so 2 questions that we've tried to address. One, what is it that maintains the sequence similarity of the multiple copies of the ribosomal RNA genes? And what are the selective forces that are maintaining 7 copies of the ribosomal operon in these organisms? So to answer that first question, Joel Hashimoto inserted a SAGB neomycin cassette into the small subunit ribosomal RNA encoding gene. And in this construct, he left regions upstream and downstream with a sequence similarity to the other operons. And he made a second construct where that SAGB neomycin cassette was such that there was no upstream region where recombination might take place. And as you might expect, when there's up and downstream regions of similarity, E. coli repairs that lesion. And the mechanism appears to be gene conversion. That is, another one of the ribosomal RNA genes acts as a template for resynthesizing this lesion. We could measure the rate in a fluctuation test at which that repair is made. And we could also see which other gene was being used as a template, because oftentimes the internally transcribed spacer would also be replaced. So we could watch that gene being repaired at a rapid rate, and the mechanism that seems most consistent with it is gene conversion. And if there's no upstream region, there's no repair of the operon. So we propose that this is a mechanism by which E. coli is preserving the sequence of the 16S and 23S genes. And in fact, then we looked at all bacteria and said, what's the relationship between the pairwise differences? So this is looking at every pair of operons in bacterium, counting the number of differences in the 16S plus 23S genes, and the distance between operons. Because gene conversion happens more frequently when operons are closer together. And that bore out in this analysis of lots of pairs of ribosomal RNA operons in the genomes that we had available at the time. So a strong, significant relationship. And in fact, that's generally the case. When ribosomal RNA operons are close to each other, they're more likely to be identical. So that answered a question about a mechanism for maintaining the sequence similarity, but it didn't answer any questions about the selective pressures for keeping 7 copies of the ribosomal RNA operon. So we went through, this is Brad Stevenson's dissertation, and decided to knock out 2 of the 7 ribosomal RNA operons. And here's a Southern digest showing that in one strain RNB is deleted, in another strain RNA is deleted, and in another strain both A and B are deleted. And we asked, well, what's the consequence of deleting some of these operons? By the way, we were smart enough to delete it so that there was no upstream region of similarity. That wasn't our first try. Our first try was, well, we'll just knock out some of this internal region. And I remember coming into the lab on a Monday morning, and Brad Stevenson, who did this work, he said, hey, I lost the knockout strain. I said, well, you want me to help you look for it? He said, no. He said it's gone. It seems to have repaired itself. And so Joel Hashimoto decided to make lemonade out of that lemon. Brad went back and knocked out a larger region, and we figured out how to prevent E. coli from making a repair. But the result here is that, so on the y-axis here is the relative growth rate of the deletion as compared to the parental strain. So a value of 1 here means that the deletion strain is growing just as well as the parental strain. So when we did these competition experiments in a minimal medium with acetate as the sole carbon and energy source, E. coli doubles with a doubling time of about 2 hours, and there was no difference between the knockouts and the wild type. But as we began to push E. coli to grow a little bit faster by changing the carbon source, in this case to minimal salts plus glucose, we began to see some discrimination. When 2 ribosomal RNAs were knocked out, there was a significant difference. And then when we pushed further by having the medium be a complex medium, lurea broth plus glucose, then in fact we could, both of the single knockouts grew more slowly than the parental strain, and the double knockout grew even more slowly. So sort of obvious, right? I mean, one reason for having multiple copies of the ribosomal RNA operon is to grow faster. Once the promoters are saturated, the ribosomal RNAs are being made as fast as possible. That limits the growth rate of the cell. So having additional copies of the operon would allow for faster growth. That was what was suggested here. And we just extended that argument to all of bacteria, saying, well, sure, the force that's pushing for the maintenance of higher numbers of operons is the capacity to either respond rapidly to a change in the environment or just to grow quickly. What was a little bit more challenging is, why isn't that always good for bacteria? Why wouldn't the capacity to grow quickly or to respond quickly to an influx of nutrients always be selected for? There are more species of bacteria for which we have a genome sequence that have 1 or 2 copies of the operon than organisms that have more than that. That's interesting. That's enough to throw you off a little bit. Yeah, well, so faster growth, more copies. That's sort of like obvious. But what's the selective pressure for maintaining few copies? I mean, maybe these are organisms that never see the capacity to grow faster. That's a possibility. But it's also conceivable that it has to do with the efficiency of the organism. So, okay, to expand this from E. coli to the rest of the bacterial world, here's the data that I think is most convincing. So each one of these points here is a different bacterial species. Mostly this is data from the literature where a maximum growth rate is reported and a genome sequence is available, so that we could plot the maximum reported growth rate versus the ribosomal RNA copy number. Notice that this is on a log 2 scale, and that the slope is approximately equal to 1, meaning that every time there's a doubling in the number of ribosomal RNA operons, there's approximately a doubling in the maximum reported growth rate. I was stunned with this result. This is, I mean, so is that really the maximum growth rate for that organism? I mean, this is just what somebody has reported in the literature. And you don't really know how hard did they work to try to get that organism to grow faster. Did they change pH, temperature, add amino acids or not? So despite the variation and despite the noisiness of this data, there is a strong relationship. So I was convinced that we were moving up the right tree, barking up the right tree, that more copies allowed for a faster and maximum growth rate. But again, that didn't help with explaining the selective forces for maintaining few copies of the operon. So we had proposed that it was efficiency. So growth rate is the number of progeny per minute. Growth efficiency is the number of progeny per mole of substrate. And the way we measured bacterial growth efficiency, first, it's the fate of this carbon. So how much of this carbon goes into biomass, and how much of that carbon is respired to CO2. So we're talking about aerobic, heterotrophic organisms. In E. coli, that number is about 50%. About half the carbon in glucose, E. coli growing on glucose minimal medium, about half the carbon goes into biomass, and about half is respired to CO2. So its efficiency is 0.5. We measured the bacterial growth efficiency of each of these strains that's highlighted here in red. So we had a spectrum across the gradient. We were being conscious of the phylogeny of these organisms and measured bacterial growth efficiency by measuring leucine incorporation and oxygen consumption. And here was the result. This is Ben Roller's, the major figure in his dissertation. And he showed that there was a negative relationship. So this is a common garden experiment where all the organisms are brought into the same garden, the same succinate minimal medium. And he measured the bacterial growth efficiency of each of these 8 different strains. The error bars here are technical, just technical variance in multiple measurements. And he showed that, again, there's a strong negative relationship. So this was the first time, this is the first indication that we have to suggest that across the bacteria there's a characteristic that's common to low copy number organisms. And that characteristic is the capacity to grow more efficiently. If that's true, we should see that reflected in the rest of the genome as well. So here is an analysis, ordination analysis. Each point reflects a genome, and the closer the 2 points are together on this ordination analysis, the more similar they are. Similar in terms of KEGG modules. So that's how we're comparing genomes. And we're compensating for the phylogenetic relationships amongst organisms as well. So this is a phylogenetic principal component analysis. And what we found using 1,147 genomes is that we could, that organisms that have lots of copies of the ribosomal RNA operon share more of their KEGG modules. They're more alike in terms of their genomes. Organisms that are on the low end of this, few ribosomal RNA operons, are also similar in terms of the genes, in terms of the modules that they contain. So we're in the process of doing this on the individual KOs from KEGG. It's a little bit more convincing, I think, by looking at and scoring modules. So some of the things that we're finding from the genome analysis is that the capacity for autotrophic growth is certainly a characteristic of organisms with few ribosomal RNA operons. That was one of the features that came out from the genome analysis. Chemotactic motility, more likely that a genome has that. The likelihood that a genome has the genes for chemotactic motility increases as copy number increases. Sort of makes sense. That's an expensive way to explore the environment, to use to chemotact towards gradients. Genome streamlining was evident both by looking at thymine biosynthesis pathways and also just the genome size. And there was more streamlining with lower copy number organisms, sort of consistent with this idea of efficiency. And PTS transporters we found more commonly as the organisms went up in copy number. And again, for each of these, we've corrected for phylogeny so that we're not being misled by lots of gamma proteobacteria having one characteristic. So here's our concept. Here's where we stand right now in terms of a conceptual model. So each of these blobs up here is trying to depict a Hutchinsonian niche, the range of conditions under which a microbe can reproduce. So this microbe here can reproduce under these conditions of pH, temperature, and resource availability. We'd need thousands of niche axes to describe the niche of this organism. Stippled within an organism's niche is its fitness. So an organism can't grow equally well, isn't equally fit at all pHs within its niche. And so when there's denser stippling, the fitness is higher. And so what we're saying is that in this part of an organism's niche where fitness is maximal, we're making predictions about that based on RRN copy number. And resource availability being one of the primary drivers of genome composition, and we're suggesting that that is predictable by this one feature of the genome, the number of ribosomal RNA operons that are encoded.
You've been fine. You got time. Keep going.
All right.
I mean, unless you want to stop, keep going.
Well, I'm just trying to decide whether to skip this slide. Now, this is a nice paper. Sebastian Bonhoeffer and Company in Science showed us where efficiency can be selected for in an environment. I don't know. I mean, I always think of bacteria that their landmark trait is the capacity for rapid growth. Maybe we need to start thinking about efficient growth more frequently. And so under what conditions is efficiency favored? So this is a simulation where in red we're always going to look at the population size of a fermentated organism, a rate-selected organism, and in blue, a respiring organism. More efficient, but it's slower because of the additional steps in the pathway. And here's one simulation. This is all in silico. A fermentative population is existing in silico here. The respiring organism is added, and it takes off and drives the rate-selected organism to extinction. So here's just one instance. And what Bonhoeffer and Company did was they looked at a number of conditions of varying resource influx and different amounts of structure in the environment. And so with a more homogeneous environment with lots of resources, rate always wins. So wherever you see red here, the rate-driven organism wins. Wherever you see blue, the efficient organism was more competitive. So as structure increases, and as the flux of resources decreases, there's more and more selection for efficient organisms. And so I think that we need to, as microbial ecologists, start to think about the selection for efficiency. We're suggesting that one way to map where an organism falls on the spectrum of efficient to rapid growth is using the ribosomal RNA operand. And if you're interested in where your organism falls, also in the spirit of the PACE lab, we've databased this information, and so we've collected RRN copy number information for all of the bacteria in the archaea, and you can just Google RRNDB and find that, and we shortly will be putting up the genomic data that is also correlated with RRN copy number. So, all right. So here's what I've tried to present today. That growth rate and efficiency vary with copy number. And I think that gives us some insights into the ecology of the organisms. And where we're headed with this is whether or not this trade-off is driven by a trade-off between the processivity and the speed of ribosomes. So K-cat, KM trade-off between ribosome as an enzyme. Genome content corresponds with copy number. We're trying to describe what those organisms look like. And then finally, good things happen when you surround yourself with smart, creative people. I learned that by being in Norm's lab and have tried to carry that on. And here's a group of people that produced the data that I showed you today. Yeah, Scott.
What is the copy number correlated to total ribosome RNA pool in a cell or total ribosome?
Yeah, good question. I don't have any idea. I don't know whether anybody else does either. Sorry?
Oh, so
I think it's the rate of accumulation that is affected.
so if you have 7 copies, is their abundance distributed over the total? Like compared to a 1 copy cell, it's probably the same amount of ribosome RNA per cell, or is there?
You're asking about the density of ribosomes in the cell?
Yeah, or the amount of ribosome RNA. Is that correlated to that?
I don't know of any data that addresses that. Yeah, yeah.
In the tomograms we did comparing sulfalotus to E. coli, and when you get with these high number of operands, the total cell volume per ribosome, there's zillions more ribosomes than you have cell volume. Sulfalotus
Per volume of the cell?
Yes, yes.
Oh, so there you go.
And
There
so
are
many,
2 points.
many of them are wrapped up too in polysomes, especially in the E. coli and salmonella. They'll be heavily packaged, incredibly efficiently packaged. So in a log phase sulfalotus, you'll have maybe 5,000 total ribosomes in the cell. And in the log phase E. coli, salmonella from Corey's lab, you'll have maybe 50,000. And they're all packaged as polysomes, all tightly wrapped together.
Yeah, go ahead.
Have you ever tried, so the operands of different sequels, have you ever tried to swap some of those around and see whether there's actually some functional
Yeah.
differences in sequels?
We have not. It's an interesting question. So it's certainly the operands that are more distant from the others where there's variation, but I don't know of any evidence that that variation has any biological significance. So you're asking whether or not all the ribosomes are the same or not. I mean, you could imagine that there are differences in the capacity to bind messenger RNA or otherwise, but I don't know of any evidence that the ribosomes are different coming from those multiple operands. They do seem to be expressed in coordination in the few organisms where people have looked. So it's not as if the organism has figured out how to turn on 3 of them under one condition and 4 under another condition. When people have looked, and that's only for a handful of organisms, E. coli, Rhodobacter, Bacillus, they haven't found that. They seem to be regulated in concert.
People look in Halarcula that has 2 drastically different RNA threads and RNA RNAs.
Yeah.
They're really divergent, right? They're 5% divergent.
That's the most divergent. In fact, that was Dan Buckley's rotation project. He had 2 fish probes, one for each of the ribosome RNA types. But doing fish in the Halarcula was that salt details. That salt challenging. So I don't know of any evidence there, but Mitch Sogan might.
Yeah, but there is an analogous situation in Plasmodium burkii.
Yes.
The gene copies are 25% different from each other, and the difference in expression depends on the merozoite or sporozoite stages.
That's the best example I know of, and the only example I know of where there's differential expression, and that's in Plasmodium falciparum. Yeah. Yeah, Lauren?
Tom, you mentioned the rectification process whereby adjacent operands can result in sequence changes or deletions. The mechanism of that.
No, we suggested that was gene conversion.
I understand
From you.
whatever that means.
There is a mechanism worked out for that, where there's a holiday junction is formed. One strand temporarily moves into the lesion and is copied, ligated in, and then a complementary strand is synthesized. So there is a proposed mechanism. There is a detailed proposed mechanism that could be tested, but I don't think that it has. We could just tell which of the other operands was used as a template, because the ITS region would move into what was being repaired, and it was different. So it wasn't just a swap of these. We now had an additional copy that had the alternative ITS region. I don't know if I explained that.
Well, I was just clicking through my brain to think how one could mess up that holiday junction and thereby prevent the rectification process.
I see.
And so I just, where did he go around? Yeah, you got any inhibitors? Probably.
No, but on this at all, San Diego State has peptide inhibitors that freeze holiday junctions.
That would be a real interesting thing to explore.
Yeah. By the way, so they were repaired quickly. Brad lost that strain after just passaging it a couple of times. Big selective advantage in LB for E. coli to have 7 rather than 6 copies.
If you look at the bacteria in the archaea that we think are non-culturable, do most of them have one ribosomal RNA, or are there some that have lots of them, which would actually suggest that we're wrong, if they're not non-culturable? There are situations where they grow really fast.
Can anybody help with that? So are there any genomes of organisms with lots of copies so you'd need a whole assembled genome from an uncultured organism and count lots of. Right. I don't, offhand, I don't, I mean, it's an intriguing suggestion that these are, they're not weedy organisms, the uncultured. So the high copy numbers are the weeds, and so maybe, in fact, that's generally true. I don't think there's data available that I know to address the question.
One of your figures that had the kind of the far extreme for those genomes that you find versus how many copy numbers?
Those were all cultured organisms. So we had to have the organism was cultured to get the genome. So she was just trying to extend that to the uncultured world.
I was wondering about, in a lot of their genomic studies that are being done now, a lot of even nominally content genomes are really small and missing a lot of stuff. And I wonder how much of the cultured organisms that we have represent only a very small fraction of what's out there, because intrinsically they're the ones that we can grow and capable of living independently, and then there'll be very big chunks of the microbial world that are capable of independent existence in the absence of their whatever species they have. But this is fairly striking, particularly at the beginning of the low nutrient environments, where you look at some similar genomes that you can do conjugates, for example, stuff from the
Right. And those are the environments where efficiency is selective.
That's right.
But
also how much cross feeding.
Yeah.
Independent.
Yeah.
Right, back to Luke.
As another way to be efficient. Don't carry all the baggage, do all the reactions yourself. Share it with.
So the interesting question would be, of the total genomes that are out there, what fraction of them are free living? It may be very small.
Yeah, that's fine, free though.
So John, if you add a ribosomal RNA up front, can you drive the growth rate up?
Well, I'd like to find somebody to do that experiment. I've been trying to do that. And actually, so I mean, initially we thought, sure, just double it and you double it. But the whole metabolism is going to be tuned to that growth rate. So I don't think it's so simple as just adding one. I think you have to add one and then wait and allow the organism to adjust the flux through all of its other pathways to accommodate that. So what I'd really like to do is to do that experiment, but put transposons on the end. So allow the organism to ramp it up, copy it as many times as would be useful, and start with something like Caulobacter that has one copy, but we can handle reasonably in the lab. And just do a Lenski style experiment where there's a homogeneous environment, selection is for growth rate, and just keep transferring it and see if the organism can respond. I can tell you that in the E. coli in 50,000 generations of E. coli, the copy number has not changed, nor has the position of the 7 ribosomal RNA operons.
Tom, thank you.